Good afternoon, dear colleagues and guests. Let me declare open the session of the Dissertation Council for the Defense of... Hello.
Yep.
For the degree of Candidate of Physico-Mathematical Sciences, Specialization 030109, Mathematical Biology and Bioinformatics, on the theme Development of Algorithms for de novo Genome and Transcriptome Assembly. According to the order of St. Petersburg University, dated the 9th of December 2019, number 12093/1, but Gordon Olgain, Doctor of Biological Sciences, Professor of the Department of Cytology and Histology of St. Petersburg University, was appointed Chairman of this Dissertation Council. Let me introduce my fellow council members. Matveeva Tatiana Valerievna, Doctor of Biological Sciences, Professor of the Department of Genetics and Biotechnology, St. Petersburg University. Unfortunately, all other council members are in remote mode. Adam Michael Filippi, PhD, National Institute for Human Genome Research, United States of America. Can you hear us? Please say hello.
Yes, hello.
Mironov Andrey Alexandrovich, Doctor of Biological Sciences, Professor, Faculty of Bioengineering and Bioinformatics, Lomonosov, Moscow State University. Andrey Alexandrovich, please say hello. And to my deepest regret, Mikhail Sergeyevich Gelfand, Doctor of Biological Sciences, Professor, Deputy Director of Kharkiv Institute for Information Transmission Problems, Russian Academy of Science Academics, is also in the remote mode today. Can you hear us?
Yeah, I'm here. I'm sorry. It was my fault that I couldn't come.
Our session is being recorded and broadcast online at the university website. The session is being translated simultaneously from Russian into English or from English into Russian. According to the federal law of the 23rd of August, 1996, number 127, Federal Law on Science and the State Scientific and Technical Policy, St. Petersburg University has the right to award academic degrees independently. The corresponding order on the procedure for awarding academic degrees at St. Petersburg University, on the basis of which we are holding this session of the Council, has been issued on the 1st of September 2016, number 6821/1, hereinafter the order. In accordance with this order, a session of the Dissertation Council is duly constituted if at least 2/3 of the approved members of the Council are present, but not less than 5 persons. Our Dissertation Council consists of 5 members. All the members are present, including, in the remote interactive mode, 3 members of the Council. Audio-visual contact has been established with them, so we have the quorum. Let me set forth the following procedure of today's session of the Dissertation Council. The total duration shall not be more than 2 hours. First, there will be Chairman's summary report on the main content of documents submitted by the applicant and their compliance with the applicable requirements. Answers to possible questions, about 5 minutes. A brief presentation by the degree applicant outlining the key points of his research, about 15 minutes. Questions to the applicant strictly on his report, no more than 2 minutes per question, and answers of the applicant, no more than 5 minutes for all questions. The requirements, the wording is very strict here. So questions is the most interesting part. So please do not take these strict words too seriously and ask your questions. Speeches of all members of the Dissertation Council in turn with their reviews, statements of their opinions, questions and suggestions to the applicant, no more than 10 minutes for each speaker. Speech of the chairman and her review, also about 10 minutes. Answers of the applicant to questions and remarks made by members of the Dissertation Council, not more than 15 minutes. Open discussion. Speeches of attendees with summary of their positions and were not members of the Dissertation Council with summary of their opinions and or suggestions to the applicant. Strictly on the topic of the study, so let us know what you think. That will be much more interesting. At that, all the speakers are kindly requested to write their names on the registration sheet and introduce themselves in full before speaking. Final answers of the applicant to questions asked, no more than 5 minutes. Speeches of academic supervisors, no more than 3 minutes. A 5-minute discussion before the open voting procedure on awarding or not awarding the academic degree of St. Petersburg University to the applicant. For the time of this discussion of the Dissertation Council, broadcasting shall be suspended. Open individual voting and counting votes by the chairman. The results shall be recorded in the minutes of the meeting. Decision on awarding or not awarding the academic degree, St. Petersburg University. Closing remarks of the applicant, no more than 2 minutes. Dear colleagues, do you have questions or objections to this procedure? No one has questions. If no one has any objections, let's start this session. Before, let me remind you to switch off your mobile phones, and thank you for your understanding. So, item 1, description of the applicant, a thesis by Przhibelskiy Andrey Dmitrovich for the degree of candidate of physico-mathematical sciences, specialization 030109, mathematical biology and bioinformatics, on the theme development of algorithms for de novo, genome and transcriptome assembly, was accepted for defense by the order of the Academic Secretary of St. Petersburg University, dated the 6th of December 2019, number 12045/1. The Dissertation Council was appointed by the order of the Academic Secretary of St. Petersburg University on the 9th of December 2019, number 12093/1. And I have already introduced to you its members, dear colleagues and guests. Andrey Dmitrovich Przhibelskiy, Researcher Center for St. Petersburg University. Andrey Dmitrovich wrote his thesis, The Basis of St. Petersburg University. He has 2 academic supervisors, PhD, Professor of Cytology and Histology Department of St. Petersburg University, Lapidus, Anna Lvovna, and Professor of San Diego University, Pevsner Pavel Arkadovich. a number of publications in which he outlines main results of his research. According to the enclosed list is 5. All the papers are published in journals included in scientific databases Web of Science and Scopus. The applicant submitted to the Academic Council a full set of documents for his thesis to be accepted for consideration and defense. All the documents comply with Clause 12, Section 3. So, I beg your pardon. I beg your pardon. All the documents are included in the applicant's file and copies available from the staff of the Irina Vasilievna Karasyova, who's responsible for today's session. Do you have questions regarding these documents? No one has any questions. Copies of documents are available from since no one has any questions. There's no need to look at the copies. Now I would like to give the floor to the applicant for his report. Большое спасибо.
Committee. My name is Andrey Przhibelskiy, and today I'm going to talk about my PhD thesis, as expected. And the title of my thesis is Algorithms for the novo, Genome and Transcriptome Assembly. And I'm going to tell what work lies behind it. Outline of today's presentation is as follows. First, I'm going to give a brief introduction about sequence assembly problem, about the Bruin graph, and a little bit about space assembler, because my work mainly implemented within space pipeline. Then I'm going to talk about expander model for repeat resolution and scaffolding, which is my first main contribution to this work. And the second part is devoted to the novo transcriptome assembly. And in the end, I will briefly tell you about my current projects and future plans. First, about the sequence assembly problem. As we all know, modern sequencing technologies do not allow us to read the entire DNA molecule from end to end. Of course, current third-generation sequencing technologies allow to sequence ultra-long reads like PacBio or nanopores. But in this dissertation, in this talk, I'm mainly focused on short-read technologies such as Illumina, and mostly I will talk about them only. And as we know, the main idea is to concatenate, to glue these reads together into an initial DNA or RNA sequence that we have sequenced. Most assemblers, they utilize the Bruin graph concept. And to understand my presentation, I would like to give a brief introduction on what the Bruin graph is. Let's have a simple example. We have a string, a nucleotide string, and we fix a certain parameter k, which is the k-mer size. We will use it for the Bruin graph. And using a sliding window, we write down all k-mers from our string. Since it's a set, we write each unique k-mer only once. And this will be vertices of our graph. Then we connect them using the following rule. If there exists a k plus 1 mer in the string, such that this k plus 1 mer starts with some prefix and adds some suffix, we name it an edge, and it connects corresponding vertices. So as an example here, we have k plus 1 mer ACG. It has prefix AC, that's a starting vertex, and it has suffix CG, that's a terminating vertex. So we have a directed edge ACG. And we do it for all k plus 1 mer in our string. Then there is a rule. If we know initial string, then we can find a path in this graph which corresponds to this string. And this is one of the most important properties of this graph. Of course, if we don't know the initial string, we cannot reconstruct it. Because as you see at this graph, we have a cycle. And we don't know how many times we should traverse it. But again, if we know the sequence, we can find the correct path. A couple more modifications to this graph. For example, we have a vertex TA. For us, this vertex is not interesting. We're interested in traversing through the graph. But this vertex has only one way in and one way out. Then we can substitute it with an edge, so in such a way so we can preserve the sequence. We have GTA, TAA, and we merge them together and get GTAA. And we do it the same way for all one-to-one vertices. And we have a nice, clear graph. And the rule here remains the same. If we know the string, we can find the path. But if we don't, we need some additional information to find this path. And the main problem here is the cycle. And the cycle is caused by the repeat. You see, our initial string has a repetitive sequence, CGT. And because of that, we have a cycle in the graph. And we cannot restore the initial sequence. That's the problem. In real-life sequencing projects, we get additional information from two possible sources. We can get it from additional long reads, such as PacBio and nanopores, which I mentioned before, or from paired reads. Paired reads are two sequences sequenced from outer ends of the same genomic fragment. And the length of this fragment is called the insert size. We have two different types of libraries, paired end libraries, which have insert size smaller than 1 KB, and we have large insert size libraries, mate pairs, which have insert size up to 20 KB. A bit about space pipeline. It's important because all algorithms I implemented within space. First, space takes reads, constructs the Bruin graph, performs graph simplification, which is removing erroneous edges. Then we get a nice clean graph, which is called assembly graph. We map paired reads back to this graph, and using these alignments, we perform this path searching procedure, which helps us to do repeat resolution and scaffolding. And we then output these paths as contigs and scaffolds. And with yellow, I highlighted models where I have contributed to space, and with reddish, orangish, I highlighted my main contribution, which is repeat resolution and scaffolding model. And the last story for the introduction is why we even started developing new assembler. Back in 2011, when we started, there were already dozens of de novo assemblers. But about 15 years ago, there was developed a new method, a new protocol for sequencing bacteria. It allowed us to sequence bacteria just from a single cell. On top, you have a coverage depth plot for E. coli genome standard sequencing data set. As you see, it is pretty much uniform along the entire genome. Below, I present a plot for single cell sequencing data. And as you see, it has very non-uniform coverage due to non-uniform amplification of DNA. At some points, you have coverage up to several thousands, at some point, about 0. And at that time, assemblers were not able to work with such data. They relied heavily on coverage depth. So we decided to develop a new tool for single-cell assembly. Okay, finally, let's move on to my work. First part about repeat resolution and scaffolding. As I said, our goal is to find correct path in the graph. It's not always possible to find one path. Sometimes it's several paths, but we aim to find correct and long paths in the graph. At first step of our algorithm, we construct a set of initial paths. They contain each path contains just a single long edge, which we consider pretty much reliable. And then iteratively we select each path from the initial set and extend it using paired reads in both directions. The resulting paths we add to the resulting set P. And we do it until all paths from the initial sets are covered with the resulting paths. Then we remove some overlaps and duplicates because we don't want to see duplicating sequences in the assembly, and output the resulting paths in FASTA format. So the most interesting part is how we extend the path. This is the most challenging algorithmic thing here. We do it iteratively. Suppose we have already a path, P1, P2, P3, and it has 3 possible extension edges, E1, E2, E3, as displayed. And we compute a certain scoring function for every possible extension edges. This scoring function uses paired reads. And we select a single extension which is good for prolonging the path. And we select extension if its score at least c times larger than score of any other extension and exceeds some absolute threshold. If we cannot select extension edge using this rule unambiguously, we stop growing path because we don't want to make a mistake and select incorrect extension. And the most interesting thing here is how to construct a proper scoring function which would give us correct extension edges. This scoring function should address such issues as, for example, insert size variation, because when we sequence paired reads, we cannot sequence them from the fragments of a certain exact size. It always will be varied. Then, as I mentioned before, we have non-uniform coverage depth along the genome because we also aim to assemble single-cell genomes. And the last challenge that I didn't mention before is chimeric repairs. These are read pairs that connect distant genomic regions and have abnormal insert size. To deal with this, we implemented a scoring function that uses read pairs connecting our path with extension edges and has insert size. Those reads should have insert size within 80% confidence interval. And of course, since we have a non-uniform coverage, we cannot rely on the absolute number of reads, but we should rely on uniformity of normalized coverage. And the last thing, in order not to make a mistake, we ignore repetitive edges in our path when we try to extend it. Unfortunately, due to time limit, I cannot go deeper into algorithmic details. They are all written down in the manuscript. So let's move on to the results. We have tested Spades assembler on multiple bacterial data sets, compared it against state-of-the-art assemblies. Here I present only a few of them, which in our opinion perform the best. And as you see, this is a E. coli conventional data set. And as you see here, we make fewer misassemblies. Misassemblies are crucial assembly errors, which we don't want to see. So the lower is better. And we have higher contiguity metrics, such as NG50 and NJ50. This is something close to a median length of our sequences we output. Okay, of course, there are many more tables, many more different comparisons, but due to time limit, I cannot present them all here. So let's move to the second part. It's transcriptome assembly. Why we started transcriptome assembly? We discovered that there are some similarities between single-cell genomic assembly and RNA-seq assembly. First, it is assembly. It requires joining reads into longer sequences. Secondly, they both have non-uniform coverage length. RNA data has non-uniform coverage depth because of different expression levels of different genes and isoforms. But there are also some crucial differences between these two problems. First, when you assemble a transcriptome, you want to assemble multiple short sequences. When you assemble a genome, you want to assemble one or several long chromosomes. Also, when you assemble a transcriptome, you have alternatively spliced isoforms, which can be very, very similar. In comparison, when you do genome assembly, you don't want to see identical sequences and duplicates. And also, RNA-seq data can be strand-specific, which can be very beneficial for our assembly. To turn Spades into transcriptome assembly, we modified graph simplification, which I mentioned previously implies removing erroneous edges from the deBruyn graph. And we substituted repeat resolution model with isoform reconstruction, but based on the same idea, path extension. Just to mention a couple differences how transcriptome assembly differs from genome assembly. We studied different assembly graphs and saw how erroneous edges in genome assembly differ from transcriptome assembly. For example, we have tips. Tip is marked here with I prime, it's a red edge. And in genome assembly, such edges usually correspond to errors in reads, and we want to remove them. But in transcriptome assembly, they may also correspond to transcripts ends and starts, and we want to preserve them. So we had to modify this procedure in order to keep correct tips and remove erroneous ones. We also have bulges. Bulges are drawn here. It's two alternative edges between same vertices. Here, the incorrect bulge is marked with E prime. In genome assembly, it's usually caused by either error in the read, again, or heterozygous mutation. In transcriptome assembly, it also can be exon inclusion-exclusion or alternative exons. So we want to, again, remove incorrect ones and keep correct ones. And we also have chimeric connections. In genomic data, they mostly appear in single-cell data. In transcriptome assembly, we found a couple types of chimeric loops, so we had to implement algorithms for them as well. And for isoform reconstruction, we used the same path extension idea, but there are a few key differences. For example, we changed overlap removal procedure because we want, as I mentioned, we want to keep similar sequences which may correspond to alternatively spliced isoforms. We also implemented algorithms that can distinguish alternatively spliced isoforms using coverage depth because they typically have different expression level. And we also implemented algorithms that work with strand-specific data. But most importantly, when you do transcriptome assembly, you can have multiple correct extensions at the same time. For example, like here, when we do genome assembly, only one extension is correct at every step. In transcriptome assembly, due to alternatively spliced isoforms, multiple extensions can be correct because they represent different exons, and all of them can be present in the assembly. So at every step, we select several extensions at a time, we create several new paths, and then process these paths individually. Again, due to time limit, I cannot go deeper into algorithmic details. So just briefly give you the results. Here's comparison of RNA space against other transcriptome assemblies. You may be familiar with Trinity, which is probably the most popular transcriptome assembly. And we, among all metrics, we think that the number of assembled genes and isoforms is one of the most important feature of transcriptome assembly because it shows how the transcriptome assembly captures genes and isoforms. Here I present comparison on human data set. So here we can measure how many known human genes and human isoforms we capture. And as you see, RNA space performs better than other assemblers. We did it on multiple data sets, human, mouse, nematoda, corn, etc. And we counted number of assembled genes on different data sets. And on average, you see RNA space, which is blue, is on top of other tools. Okay, that's it about my work. Just a summary now. During my work, I developed repeat resolution and scaffolding model, which can work with both paired end and mate pair reads and can address the problem of non-uniform coverage. I developed RNA space pipeline for transcriptome assembler, and as we tested in our lab, the designed assembly shows superior quality to other tools. The work, as mentioned, was published in 5 papers and presented in 3 international conferences. Finally, about my current work, as the whole field of transcriptomics moves on to long reads, I also start working on long reads, such as nanopores and isosec, and I work on both de novo assembly and reference-based analysis of long reads. And of course, since they're longer than Illumina, they can be very helpful in quantifying different isoforms, discovering novel isoforms, and especially like single cell and spatial transcriptomics. That's it for now. Thank you very much.
Is there any questions, please? Вопросы? Ну, конечно. Все молчат, как партизаны.
Everybody is silent.
Okay, should I have a number of questions?
У меня есть вопросы.
Should I?
Hello, can you hear me?
Yes. Should I?
Yes, we can hear you. Please ask your questions.
Okay, so, well, I'll refer to pages of the thesis rather than to slides. So, at the very beginning, at one of the...
Так, хорошо.
Подождите секунду, сейчас слышно
Говорю
плохо, то ли
медленнее.
потому что синхронно идет какой-то, я не очень понимаю.
Говорю медленнее.
How does the quality of a transcriptome assembly transcript assembly descend on coverage level? Hello, did you hear the question? Could you please repeat your question? I did not understand the question. I will ask again. It's obvious that some genes have quite high coverage. Some genes have low coverage level. How does quality of assembly depend on coverage level? Could you all hear the question? This is a very interesting question. We studied this factor. We used simulation data where for each gene we know coverage beforehand and how our assembler copes with assembly depending on coverage. Unfortunately, I don't have a slide for this. This information is available in my thesis. The wider, the more coverage we have, the more genes. Coverage 5 assembly is impossible except for some very simple questions. Over 5, then it's possible to assemble, but not all the tools are equally efficient. Our assembler performs better for genes with low coverage and assembles them better than other products. Thank you. I have one more comment to curator. Sometimes too late now. There's no pointer. If no one has any questions, let us proceed to the next point. Reviews.
Sorry, I have a couple of questions.
Михаил Сергеевич имеет вопрос.
Есть?
Да, у Михаила Сергеевича есть.
Пожалуйста.
Yeah, please go on.
Миша, да.
If you think that I'm in Moscow and because of that will not ask questions, you are wrong. Okay, so it mainly relates to RNA spades. For instance, you say that you collapse bulges that consist of edges with similar length. Does it mean that you would collapse mutually exclusive exons in many cases?
Yeah, this is a very good point, and yeah, when we
This is it.
really kind of missed this point, but now, after Mikhail Sergeyevich suggested this, we really modified algorithms for bulge removal. So now we do bulge collapsing based not only on length or coverage, but also on sequence content. So if two alternative edges have similar content, sequence content, they differ, like, for example, 1-2 nucleotides, most likely it's either mutation or even more likely a sequencing error. So we collapse those. But if sequences are different, we don't collapse bulges even if they have equal length. And because this is, as mentioned, could really correspond to mutually exclusive exons. And yeah, thank you very much for the suggestion. In previous version, we don't have that. Now we modified the algorithm. Thank you.
Okay, cool. Then you say that chimeric junctions, that if two isoforms have long constitutive exons, so there is a long insert, then you have a long constant region which is longer than the insert size. It's clear that you cannot resolve the alternative events at the ends of this constant region. But then you say that the use of expression data might help because you would have correlated coverage in these two distant alternative regions. But that implies that there is actual long-range correlation between the selection of elementary alternatives, which might not necessarily be true. So could you comment on that?
This idea, it works, but of course it's not works for all cases, because first of all, as you correctly mentioned, we may have quite non-uniform coverage along the transcript. In this case, we would probably not resolve these alternative events and just stop. And the second case when we have more than 2 alternative isoforms within the graph component. That also is not the case we can resolve. So yeah, the idea is pretty limited, but we try to get out of the coverage values as much as we can. So yes, this algorithm works for rather simple case when we have 2 alternative isoforms with very different coverage, like really different. We have a pretty high margin between low and high covered isoforms. And when alternative exons, the coverage of alternative exons really do correlate. So yeah, of course, the application of this idea is pretty limited, but as we analyze different graphs and we see at least sometimes it works. Hopefully more, but we cannot be sure how many times it really helps us.
Okay. Well, I actually have had a couple of more questions, but they are pretty technical, so I think I'll save time and would not answer them. They are really technical. They are listed in my review. So, okay, well, basically I'm done.
Okay, but I think I have answers for at least some of them, which I...
Yeah, but they're minor. I mean, I'm okay with them. Thanks.
May I also ask a quick question? Andrey, could you please go back to the benchmarking slide with the RNA quast metrics and just give me a quick description of how those are computed, how is a misassembly defined and so forth. I'm not familiar with RNA quast, so I was curious.
Yeah, of course, it's not that, I think, straightforward as in Genome Assembly, because I think it's, well, I would say it's pretty easy to show someone what misassembly in Genome Assembly looks like. So what we do is we map assembled transcripts onto the genome, and we compare these alignments versus annotation file versus known genes. And we consider a misassembly as following. If a transcript maps, like assembled contig, let's say, maps to different genes, and also we use, and this is done by some spliced aligner, and also we use simple blot to map on the transcriptome. And if we see that same contig has mapping to different genes, both in the genome and in transcriptome, we say this is a misassembly. Like, it is confirmed by 2 different aligners, both spliced aligner and simple blast mapping to the gene database. Duplication ratio is a bit tricky because, of course, we have alternative isoforms and so on. So when we do this quality assessment, we assign each contig to a specific isoform. And if we see that 2 contigs are assigned to the same isoform and they do overlap, that contributes to a duplication ratio. We don't want to see a lot of contigs mapping to the same isoform. Database coverage is pretty much simple. It's just percentage of nucleotides covered.
And is the database coverage relatively low just because there's a lot of unexpressed transcripts, that your reference includes everything, but your data set doesn't include all of the transcripts expressed?
Yeah, correct. Database includes all known genes, all known transcripts, which we have in GTF. Of course, when we were publishing a paper, we presented not simply number of like database coverage or number of assembled transcripts. We first calculated the number of what we actually might see in these data sets, because of course this number will be low because, yeah, it's normal, I think, for a human tissue to express like 10 to 30% of the genes. Well, and the number of assembled genes, the isoforms, we assign a contig to it. Isoform, and we see what is the percentage of nucleotide covered. If it's higher than 95, for example, we say that this gene or isoform is 95% assembled.
Good, and then I'll just ask one last question about the long-read RNA-seq. How complete have you found, say, the human reference annotation to be with regard to alternative splicing in isoforms? Have you looked at any of the long-read iso-seq data sets and found that the annotation is sufficient? Or is there room for improvement?
I cannot say that I personally studied a lot of alternative isoforms, but I worked with a number of both iso-seq and direct nanopore sequencing data, mostly from mouse and human, and mostly from mouse and human brain. And as we see, there are a lot of cases when we see novel alternative isoforms, alternative transcription, start sites, and so on. So as far as I know, there is a lot of room for improvements, and the group I'm working with, they are currently trying to make a special annotation, like specifically for brain regions, like for isoforms that express particularly in brains, in different brain cells, like neurons, astrocytes, and so on. So yeah, I think there is a lot to do in the annotation.
Good, thank you.
Thank you.
Микрофон включи.
В том порядке, в котором...
Let us present the reviews. And I'm going to give the floor to Andrey Alexandrovich. And what should I do? Please read your review. Read your review. Reviews, please. I see. What else do I have to say? Since reviews have been published at the university website, they are available. So it's possible to read or only give questions to the applicant, of course. So you can make it shorter. So well, I will not list my technical comments about the structure and style of the thesis. My comments and suggestions I'm going to give now. First, the algorithm has a number of significant limitations when applied to eukaryotes. Can you hear me? Yes, we can. And there are such many eukaryote genomes, as there are also polymorphisms, so there is a question about assembly in case of it's polymorph. Shall I ask all my questions, or shall the applicant answer each questions one by one? Okay, I will answer each question. Speaking of genome assembly, I will answer in Russian. Answering the same. Speaking of genome assembly, when space, of course, we had ideas to assemble large genomes, but since we failed a number of times, so we stopped working on this. Because spades is not good for eukaryotes. We work only with small bacteria assembly, and we advise our users advise them against using for eukaryote assembly. As for so the polymorphism is not that pronounced. Only we may run into bulges, which I mentioned already. And so we will. Second question is more a suggestion, but still, in assembly, in transcriptome assembly, you use only experimental data. However, or maybe in most cases, there's a large amount of data of related organisms, known references, and known genomes. And I think that if you managed to introduce functions for analysis for related organisms, the quality will be better. And coverage requirements will be lower. Yes, this is a very good comment. Thank you. Yes, at first we worked on assembly, but now we have a hybrid assembler, and then long sequences, and in the future maybe we will use database of related organisms. This is a very good idea, which I think we should add, because our algorithm, we have some ideas, since we can use long sequences, or why not use similar genes, because as far as I know, such software is already available, not only reads, but existing bacteria database, and it helps them a lot. And assembly, the quality of assembly is much better. So that's one of the future spheres for the research. Thank you for this comment. And next is both a comment and a suggestion. You see, algorithm suggested based on the use of support functions, and when discrete or discrete values 0 or 1, and maybe this is not very good to use such value. If you use a valued graph technique, would be better, the reliability will be higher, especially yes, indeed, at some point we had such ideas, but at that time we thought we should quicker create some reliable heuristic. Yes, we had ideas to maybe about learning, and we tried different possibilities and not all the ideas which we considered, which we did not include. Maybe I didn't include all the ideas in the text. This is a good idea, but maybe it would be best to say that we didn't have enough time for that. Well, thank you. Other comments are all editorial. It's about misprints and inaccuracies, so maybe it's not necessary to mention. So I will probably, that's it I wanted to say. Thank you. And you have to say the obligatory part. Let me that everything complies the thesis of Andrey Dmitrievich Przhibelskiy on development of algorithm for genome and transcriptome assembly corresponds to the basic requirements set by the order, the 1st of September 6821/1, order awarding academic degrees at St. Petersburg University. And the applicant reserves the academic degree of candidate of physical mathematical science, specialty 030109, mathematical biology and bioinformatics. Paragraph 11 of the has not been violated. That's all. Thank you. Mikhail Sergeyevich, now it's your turn.
Okay. So basically,
Миша, вы нас
the thesis
слышите?
combined. Yes, I'm trying to say
Ваш
something.
отзыв, пожалуйста.
That's exactly what I'm trying to do. Hello?
Yeah, we can hear you. Please go on.
Okay. So it's something with the translators booth probably. So there are two main contributions in this thesis. One is a set of heuristic improvements to already existing program space, and the other one is a description of RNA space, which is the brainchild of the candidate and it's his main contribution probably. Actually, both chapters, chapter 2, which describes this repeat resolution modules for space and some other minor improvements, and chapter 3, which describes RNA space, are equally important, and both are very good contributions. So I have, I've had my questions, but I have no criticism on the substance. On the other hand, I've had considerable criticism on the writing itself, because I have a strong feeling that the applicant, well, the candidate, quite naturally probably, but still wanted to spend as little time as possible on thesis writing. And sometimes it's okay, but in this particular case, it resulted in two problems. First of all, there is no good review, and all information about other programs or other like predecessors or what's on the market is rather patchy. And secondly, the exposition itself, especially in chapter 2, is not very logical. Sometimes terms or notions are used before they are defined. Sometimes the reader is referred for details to the author's publications, which is actually contradicts the very idea of thesis as a self-contained text. So I have my criticism on the form, but that given aside, I think it's good work. It's quite, the list of publications is quite impressive, and the author is, the candidate is the first author, and the main publications related to this thesis. I think that the scholarship of Andrey is excellent, and the results are original and important, and they are clearly relevant for the science as it's now. So the algorithms have been implemented in popular software. The papers are well published. The results have been reported at major conferences. The papers are also have also gathered a considerable number of citations because the software is widely used. So I think that the dissertation meets, well, the formal part is that the dissertation meets all requirements of the St. Petersburg State University and the Russian Foundation, and Andrey Przhibelskiy should be awarded the degree of candidate of physico-mathematical sciences in mathematical biology and bioinformatics, specialty 03.01.09. And by the way, I have noticed a misprint in my review because in the written session, written version, I see physico-biological sciences. It's clearly physico-mathematical sciences, a degree that Andrey is seeking. So that's it. I think it's a very good research.
Thank you.
Okay. Yeah, I'm done.
Well, I guess Mikhail Sergeyevich asked all questions after the talk. So just a minor comment. Yeah, I agree that the text
is a minor.
Will not say anything else. I think now I can only use this comment in future when I will write, I don't know, reviews, and I can pass it to future generations who will probably defend soon. Thank you very much.
Okay. Adam, now it's your turn. Are you ready to...
Dr. Filippi, it's your turn to present your review, please.
Yes, can you hear me?
Да, вы меня слышите?
Please.
Good. The other reviewers have already stated many of my comments, and I'm in large agreement with their impressions of the work and the thesis. I think these are very important and difficult problems that Andrey has worked on. I want to congratulate him on the good work that he's done. I remember meeting him at a meeting in Boston in 2014 and seeing this expander work first presented. And I think that is a key component to some of the success Spades has enjoyed over the past 5-10 years. The RNA-seq work is exciting. I would have liked to see a little bit more exposition of that in Chapter 3. As was stated, some of the references went to other papers, and I thought it could have been included in that chapter, so I didn't have to look up those other papers to understand the validation in particular. But nonetheless, they are published. It is good work. Andrey has demonstrated his scientific independence and creativity, and the dissertation meets the requirements, and I support granting the degree. Good work, Andrey.
Thank you very much.
Tatiana Valerievna, now my task is very simple because many issues such as relevance of the work has already been discussed, and is beyond any doubt. I had include some comments, but they are mostly editorial, and that is why I would rather comment on the focus on the strong points, except for relevance of the topic. I'd like to mention good style of the work when someone starts the basics and from molecular biology, algorithm solutions, and then proceeds to his discussion of his results. So it's very easy to read this thesis. Another strong point which I would like to mention is good conclusion in which the author not only summarizes his work but also speaks about the prospects. And speaking of prospects, since Andrey is working with repeated sequences, I would like to emphasize that it's very interesting. The assembly of long inverted repetitions is also a very challenging task because the problems that may occur during biological stages of sequencing should somehow be compensated computationally. So in this sphere it will be interesting to develop. So I was very impressed. My impression is very positive, and in conclusion I have to say the formal part that thesis by Andrey Przhibelskiy, development of algorithm for genome transcriptome assembly, corresponds to the basic requirements established on the 1st of September 2016, 6821/1, on awarding academic degrees at St. Petersburg University. And the applicant, Andrey Przhibelskiy, deserves being awarded a degree of candidate of physical and mathematical sciences, specialty 030109, mathematical biology and bioinformatics. Clause 11 of the above-mentioned order has not been violated by the applicant. Thank you very much. And it's my turn now. And I am one of the users of software produced by the applicant, and I will not give you any excellent any praise, so I'll ask some questions. In the text of the thesis, emphasize the importance of choosing the correct length of chimer when assembling, and the method for determining the optimal value for chimer is described in detail for transcriptome assemblies, but no similar description for genome assemblies. How were chimer lengths selected for genome data assembly? My user impression is too long 21, 33, 55. It should be too long. Usually we use shorter lengths. Shall we answer questions one by one? Okay, let me answer. This is also a very good question. I, maybe deliberately or on purpose, I think it's not directly connected with the theme of my thesis. How did we choose optimal values for K is a K-mere length is a long story. We entered different data, so it's hard to identify data that yields optimal values. Comparing assemblies, we used Quast. Was my colleague presented here a little over 1 year ago. So in the end, we started using K values, which yield good results. An important point here is that in reality, the size of K-mere only affects the graph itself, how it looks, what it looks like. After the graph is ready, we're still looking for paths. It's not that important for K-mere to be ideally selected. Algorithm for path selecting paths can produce good results. Even K is not ideal. What's important is to keep it in the reasonable range. That's my answer. Next question, which I asked to the text of the thesis. One of the key algorithms developed by the author is the algorithm for resolving expander repetitions. But in the thesis, there's a detailed description, but there's no graph. So I think I saw it in your presentation. Can I see it again? Yes. There was detailed description closer to pseudocode. Yes, this was a serious omission after I read. So I explained how XSPADE worked and gave other graphs, but I didn't give pseudocode and how my algorithm work. And well, this is a very good comment. In the future, if I talk about this, I should remember this in the future. But what was mentioned? Yes. Yes, I included this slide in my presentation today. The thesis briefly describes how SPADE resolves tandem repeats. And you also say that in assumed number of copies greater than 1, the algorithm inserts sequence of n characters, does not permit repetitions accurately. Can your method be improved using other types of sequencing data? It's very important for me because I'm working on tandem repetitions. Also a good question. With tandem repetitions, we were struggling. Here we have Sergey, with whom we discussed this many times, and in the end we decided to say that it's better to say we don't know how to pass a repetition correctly. Let's leave those n symbols and let's leave it up to the user, and we want the user to be aware that here we cannot produce accurate number. So if we don't know the accurate number, we don't want to mislead our users. We shall tell our users that we don't know case sets to identify the exact number of copies of tandem repetition. So of course we could use more aggressive strategies that we used in the past, but we decided not to do this. So yes, this is a good question, especially considering how this assembly is, the technology is developing, and I know that in some regions researchers are struggling with this issue very hard now. One more important question. Chimerical edges called studs. Is there a better term? Because the term start is already used for something. As pin is a secondary structure of RNA, and they have nothing to do with ages. Maybe you can think of another term. Yes, the term is bad because indeed RNA already has pins and is used quite common. I don't know if is it necessary to find a special term for such a small thing as chimeric ages. All my other questions do not require answers because all types of way on which the algorithm was tested, summarized as short Latin E. coli and H. sapiens require no explanation. Some bacteria I was not able to recognize, and in addition I found 2 misprints, and otherwise the thesis is extremely well written. And before I say the obligatory phrase, I'd like to say something informally. The thesis and today's presentation, we here struggle with repetitions, which gets in the way of our research, but I am positive that next age of biology and next task will be figuring out repetitions which constitute the major part of genome, and 20,000 genes for in humans, and is not enough to create something without repetitions. Andrey struggled with this enemy, and now you have good understanding of this issue, of this problem. So he's helping us, he's giving us a hand. Now I have to say that the thesis by Andrey Przhibelskiy on development of algorithms for genome and transcriptome assembly corresponds to the requirements established by the order number on the 1st of September 2016 6821/1 on the order of awarding academic degrees at St. Petersburg University. And the applicant, Andrey Przhibelskiy, deserves to be awarded the degree of candidate of physico-mathematical sciences, specialty 030109, mathematical biology and bioinformatics. Clause 11 of the above-mentioned order has not been violated by the applicant. Well, now, do attendees have questions or comments? Silence. Then let us proceed to next clause, speeches of the academic supervisors. Alla Lvovna, welcome. Please stand here. I have 3 minutes, and I will try to stick to the time frame after such reviews, after such good words. Yet I would like to say high quality of the work and elegance of solutions. Struggling with repetitions is hard, and not everyone can find a solution. Here a solution was found, was very elegant. Also the independence of the thesis and ability of the applicant to work in a team. He's a team player, and he can also work individually. And a big part is a very essential element of the work. Speaking of repetitions, I think in nearest future, this struggle will lead us to a victory, and we will soon come back here for presentation of doctoral thesis by Andrey Przhibelskiy. Is that all, Alla Lvovna? Thank you, well done. The second supervisor is not present here today. That is why I will read his. The second supervisor is professor of California, University of San Diego, USA, a PhD of physics and mathematics, senior advisor, Center of Algorithmic Biotechnology, St. Petersburg University, Pavel Arkadievich Pevzner. And the key points of his review. In the course of his work, Andrey D. Przhibelskiy demonstrated important ability to conduct scientific research independently and to use scientific literature in the field of bioinformatics and to interpret the results obtained from the analysis of biological data correctly. Developed and implemented in the process of his work computational methods and algorithms testify to deep knowledge of the author in the field of computational biology and computer sciences. It is also worth noting good ability of Andrey Przhibelskiy to present scientific results both in written texts and spoken reports. Results obtained by Andrey Przhibelskiy in the course of his work became the basis for further developments in the laboratory. Thus, various algorithms implemented in the framework of Genome space assembler, including expander module, have been repeatedly reused in various versions of SPADES, such as hybrid SPADEX, meta SPADEX, etc. The creation of RNA SPADEX further, it became the basis of transcriptome direction in the laboratory of the Center for Algorithmic Biotechnology, in the framework of which methods for analysis of new transcriptome data are ISO-seq, ASP, ISO-seq, etc. are being developed under supervision of Przhibelskiy. The main results presented in the thesis have been published in 5 articles, journals indexed by Scopus and Web of Science, and have a high citation level. In addition, the results were presented at important international conferences on bioinformatics, in the form of spoken and poster presentations. According to relevance, volume of obtained results, scientific and practical significance, a thesis by Andrey Przhibelskiy, development of algorithm for assembly of genomes and transcriptomes, submitted for the degree of candidate of physical and mathematical sciences, meets the requirements for the thesis for the degree of candidate of sciences and can be accepted for defense. Next, according to our procedure, we have to discuss, but I don't see any point for discussion here. Dear council members, do we need any time to discuss? Do we need time for discussion? It's not clear what we should discuss. There's no point for discussion. Do you have questions? No, we don't have. Yes, I also have this feeling. Okay, so we don't need any time for discussion. Then it's time for the open voting. And we should if we disabled broadcasting, but we didn't do this, so we don't have to resume broadcasting again, then I put the question of awarding to Andrey Przhibelskiy academic degree of candidate of physico-mathematical sciences, specialty 030109, mathematical biology and bioinformatics, to the open individual vote. Let me remind you that a decision of the Dissertation Council on awarding academic degrees shall be considered positive if more than a half, but not less than 3 members of the Council taking part in the session, according to Article 23 of the procedure. Then I have a list. Matveyeva Tatiana Valerievna, your opinion? I am all for awarding. I support the decision. Thank you. Andrey Alexandrovich Mironov, your opinion, please. Deserves awarding the academic degree. Mikhail Sergeyevich Gelfand, your opinion?
For. Award the degree, yes.
Двумя руками за. Спасибо.
With your both hands. Thank you. Adam, what is your opinion?
I also support awarding the degree.
Thank you very much. Ну и я саму.
And I'm also for awarding the degree, counting the votes and announcement of results. So, dear colleagues and guests, I'd like to announce that out of 5 members of the Dissertation Council present at the meeting, 5 persons voted for, no one voted against, and no one abstained. Thus, the decision to award the degree of candidate of physico-mathematical sciences specialty 030109, mathematical biology and bioinformatics, has been made. Now, the candidate of sciences is given the floor for his closing remarks.
I want to acknowledge a lot of people who participated in this defense. First, of course, committee and all my reviewers. Thank you very much for your work, for your time that you spent on the reviewing my text, which, as I know, is far not perfect. And I don't know, I think words cannot describe how much I'm grateful for your contribution. Of course, I want to thank all organizing committee, translators, technical staff, and people who helped me to organize all this event, because it also is a huge work. Indeed, my scientific advisors, Pavel Pevsner and Alla Lapidus, I'm very grateful to them as well, because they were very patient at some moments when I was not delivering as expected. But they waited, and finally here I am. Of course, I want to thank all my colleagues, because all I described today is a joint work with the different people from my lab, and some of them are here today, and I am very, very much, I appreciate their work, and of course, without them, I think this work would not ever be possible. And okay, finally, my family and my friends who were supportive at some moments, but in general, I think without them, I wouldn't be here as well. Thank you very much. That's it.
That's it. Заседание объявляю закрытие.
That's it. I announce the session closed. I thank everyone for participation. Please disable online broadcasting. Thank you.